Good afternoon, everyone. Today I'll be presenting on the work I did for my master's, which the topic was biodiversity monitoring for rocky shores in KwaZulu-Natal. I did my master's under the supervision of Dr. Angus MacDonald and Dr. Maya Faff. As you know, constant monitoring of systems is required in order to ensure that we always have the most recent information on systems, and this ensures that we are able to manage these systems since we have information that represents the system as it is at the current time. Now, the last comprehensive study on the KwaZulu-Natal coastline was a study done by Dr. Kerry Sink in 1996. So we felt as though there was a need to revisit the system so we can ensure that there is an updated format of the state of the system. So the reason why we're looking at rocky shores is because rocky shores are very diverse environments, especially the KwaZulu-Natal rocky shores, which are a transition point between the tropics and the temperate region. So they have much higher diversity. The rocky shores, even though they are very diverse, are also undergoing a lot of pressure because they are very easily accessible to humans. So the aims of my study are then to characterize the changes that have occurred since the study that was done by Dr. Sink in 1996 by resurveying 14 of the 39 sites that she surveyed back in 96, and I compared the species richness between 1996 and 2018, and I also compared the changes in community structure. One of our biggest limitations with the study was that the sites, the zones in the previous study were not fixed. Therefore, we had to rely on identifying the dominant species that were characterized by Dr. Kerry Sink to describe each zone. We then separated the intertidal zone into 4 different zones: the top shore, the high shore, the mid shore, and the low shore. In the 2 upper zones, the top shore and the high shore, we took 10 quadrats in each, and in the 2 lower zones, we took 20 quadrats in each, and that gave us 60 quadrats. Within each quadrant, we took percentage cover of the sessile species, and we also took counts and measurements of the mobile species. We then used the counts and measurements of mobile species to give us an estimate of percentage cover of the mobile species. When we compared the species richness, we found that there was an increase in species richness from the 1996 study to the 2018 study. As you can see, the dark gray blocks represent 1996 and the light gray blocks represent 2018, and the asterisks are an indication of significance. With the species richness, we also found an increase in evenness, especially in the lower zones. The upper zones, the top shore had a decrease in evenness, and in the high shore there was no change in the evenness. When we explored the community structure, we noticed that we found some trends that caught our attention. And one of them was, if you take a look at the top shore, firstly, the colors indicate different years, and the shapes indicate different sites, and the blue vectors indicate the species that contributed the most to the change. You will notice in the top shore that periwinkles were one of the species that actually contributed the most to the change, but they're showing a decrease in number. This is concerning since periwinkles are the species that are characteristic of the top shore, and they have been used in other studies to as indicators of change because they are exposed to the atmosphere for a very long time, and they are then again exposed to water at high tide. So when we noticed that they had decreased, this was something that was concerning. You also noticed in the low shore there was a decrease in the mussel biomass, and this was also something that caught our attention because mussels are very important for the livelihood of communities. When we further explored the species contributions by site, we noticed that there was a decrease in mussel numbers along the entire coastline. Just as a key to this table, the blue circles indicate increases, the red circles indicate decreases, with the size of the circle indicating the magnitude of the change. The smaller circles are between 5 and 9%, and the bigger circles are between 10 and 20%. So there was a between 10 and 20% decrease in the mussel biomass along the entire coastline. Now the question that we had then was, what was causing the decreases in mussel biomass? What we found is there is a lot of harvesting on the KwaZulu-Natal coastline, since some of the coastal communities still depend on shore resources for their sustenance. In the top picture, there is a picture of the Mapelan site, where harvesting usually happens every season. And the other biggest challenge with this particular site is that along with the harvesting impacts, there is also sand loading or sand inundation. And this further aggravates or increases the effect of harvesting, as whenever juveniles come in to re-recruit, to re-establish the community, then they get smothered by the sand. And this then has a negative feedback on the system. Another alarming discovery that we made while we were exploring the mussels in the low shore was the presence of 2 invasive alien barnacles. Dr. Maya Faf, whose presentation will follow mine, will go into further details about which steps we took after we found these. In conclusion, we did note increases in species richness, which we believe was due to movement of alien species into the KwaZulu-Natal coastline, and this combined with loss of the native species has resulted in an even distribution of in the lower zones, because usually the native mussels, the perna, tend to dominate and cause a low evenness. But with the loss of them and the moving in of another species, the community has become more even. And even though some increase in species richness was observed in the upper zones, we found a decline in evenness. And this we believe, was caused by pressures such as sand loading and also climate forcing on the top shore. What we recommend is that further monitoring be done on the system, and it needs to be done more frequently than at every 20 years. And this monitoring, for this monitoring to be done properly, we then have to establish, for this monitoring to be done easily, we need to develop a monitoring protocol that will be both easy to use and not too time consuming, which is what the second chapter of my MSc focused on. Thank you.